the major bottleneck for tracking COVID-19 infection is indeed the possibility of performing a large number of tests. And let's say the common point is that, of course, we need to count with performance methods presenting an enhanced sample to answer speed, so it needs to be quite quick, reduce reagent costs, and that's actually a major issue, I guess. An easy way to make them available, because large instruments can be a problem, and the acceptable sensibility of these methodologies. In addition to the different methods that are already available, today I'm proud to present this methodology called PostCoV, which is actually aiming to combine pooling strategy, so pooling candidate samples together with massive parallel DNA sequencing as a way to perform COVID diagnostics. Pooling strategy has been already used in the past in the context of RT-qPCR. So the idea being that you can combine multiple samples, and if you have a positive hit, then you have to repeat again this assay until you basically detect the person that got infected. So this is a way to reduce costs, but the problem is that, of course, you need to repeat until you find the contaminated person. So to solve this problem, recent reports from the US mainly combining this strategy of pooling with the use of molecular barcodes and massive parallel DNA sequencing. And actually, as you can see in this preprint of June 2020, the authors claims that they could even combine up to 100,000 samples. So we are talking here at real massive screening for COVID-19 samples. But the problem is that if you want to do so, you need to count with these expensive Illumina next generation sequencers. And of course, if you think to deploy this in, let's say, in poor countries or in different parts in France even, this becomes a problem because we don't have sequencing centers all over the regions, right? So what I propose today is to take advantage of a cheaper solution, which is actually the use of the Oxford Nanopore technology. So Oxford Nanopore, as you may know, is actually a reduced scale sequencing technology, which actually allows to basically it's a portable solution, as you can see in this figure on the bottom. I don't know if you can see it. So the sequencer is as small as a pen, and the instrument is also quite cheap. So basically, we are using this technology right now to basically develop this COVID-19 diagnostic. So our preliminary data are demonstrating that we are able to detect as few as 100 copies of this COVID-19 viral material. In addition, we have recently shown that we can pool more than 100 samples in once. So basically, we are able to combine multiple samples and still detect the viral sample. So, but of course, now our ambition is actually to go beyond. We are trying to upscale to even 1,000 samples. That's actually one of the points that we are trying to address. But keeping in mind the current needs in tracking these variants that everyone's talking about. And because we have the chance to access to the genome, now we are modifying our protocol such that we could actually trace the virus sequence diversity at the level of the spike region at the same time that we perform the diagnostics. So the idea being behind that we could diagnose and at the same time check whether we have particular mutations within the spike region and all in the same assay. So basically, that's actually the focus of the project I'm working on, and I'm actually quite thankful to DIM ELICIT because they are supporting this effort.
So I think everyone is convinced that detecting antibodies production is key for several assessments, for instance, for diagnostic, but also to assess the presence of protective antibodies titers upon vaccination, as well as the seroprevalence assessment at the population level. And since several years, there is several analytical tools that are able actually to detect antibodies, such as the famous ELISA test, but also some rapid detection assays that allow point of care. And for all best techniques, actually there is a common feature is that you really need to find a good synthetic antigen, and you need to produce it. You need to purify it. And there is something that is also key and important, is that you need to covalently graft it on the surface or on nanoparticles. In that context, actually, we propose another technology, a new technology that actually used bacteria to display the antigen of interest. So our idea is to directly transform bacteria with the plasmid, the synthetic antigen, and the bacteria will export the produced protein on the bacteria, on the surface of the bacteria. So it's a one-step process. You don't have to purify the antigens. It's also low cost in a sense because you can produce a lot of bacteria in bioreactors. And also it allows a fast reactivity. In, for example, facing appearance of variant, you just need to order another plasmid that will code for a specific antigens. And this will allow you to after overnight productions, to have a bacteria acting as a colloidal particles with a genetically encoded antigen on their surface. So we also combine in the lab the possibility to use these antigen bacteria and to manipulate them in space. In red here, you have the antigen presenting bacteria that are magnetic. So if you just put a magnet at the vicinity of your sample, you are going to highly concentrate the antigen presenting bacteria. And I'm showing you now a proof of concept where we have actually, on the picture on the left, we have the accumulation of the red bacteria that are presenting the antigen and that can magnetically capture the target, the antibodies. We can also see that if we put on a tube the antigen presenting bacteria in presence of the antibody, within a couple of minutes, we can see a pellet. So that's the red circle on the bottom. So in the context of the SARS-CoV-2 system, actually, we decided to target the phosphoprotein N. And what we did is that we engineered several variants. So we made some protein engineering, and we decided to focus on the RNA binding domain, as well as the dimerization domain of the N protein. And I'm going to show you now our preliminary results. What we did to demonstrate this proof of concept is that we incubate our proteins, our bacteria that are presenting the N protein on the surface, and we mix it with antibodies raised against the N protein that were fluorescently conjugated. And what you can see here on the top right panel is that it's single bacteria. And we can see that we labeled the surface of the bacteria, and it's highly efficient. And so we are very happy here. So we can have actually a bacteria that display the phosphoprotein N protein. And now what we are going to do is to assess the specificity and the sensibility of the assay using ELISA test, agglutination assay, as well as lateral flow assay. So thank you for your attention.